Hey guys, how you doing? So welcome to chapter 14. In this chapter we're going to focus mainly on chromosomes and sort of how they are connected to genes. Okay, let's jump into it. Okay, now chapter 14 is a fairly short chapter, and you guys are probably already aware. Here are the main information about it. Okay, I'll let you guys read through it, but let me just sort of go through some of the definitions. Now you've heard some of these words before, but I'm just going to sort of reiterate them a little bit again so that you understand what we were referring to when we're talking about genes and chromosomes. Okay, so your regular cells in your body that is not a gamete are called somatic cells, and somatic cells are characterized by having a diploid number, a full 2n number of 46 chromosomes in humans. Okay, that 2n indicates a diploid number, whereas gametes are sex cells such as the sperm and the egg, and they have a haploid or a singular n of 23 chromosomes. Okay, here is a human karyotype for males. There's the line between them there, and here is one for females. As you can see, the difference between them is that males have an XY and females have an XX. All the other chromosomes are set in these kind of pairs, and you can sort of see they're arranged according to size. Okay. Now, there's no correlation between chromosome number and complexity of an organism. Yeah, it just contains the genes that it has, but as you can sort of see there, some of them will have more or less than humans, but none of which are comparative in terms of their intelligence level to people. Same can be said of plants. Some plants have a lot of chromosomes, but don't, are necessarily not any different from any other plants, or in terms of complexity or anything like that, right? And so complexity of an organism isn't tied to its chromosomal number. Now, to understand chromosomes, we need to understand how the DNA is structured. So a single double helix strand coils together to form histones. Histones are these little cookie-looking thingies here, but they're basically wrapped around itself many, many times. Now, histones will then clump together to form nucleosomes, and these are the nucleosomes there, which will then loop together to form your chromatins, which will then condense to form chromatids and into one chromosome. Now, chromosomes join together at a particular location called the centromere, right? And the centromere is that middle region that joins the two chromosomes together, and the top arm is called the P arm, which is sits at the top, and the bottom one is called the Q arm, which sits at the bottom. Okay. When we're looking at images and we're looking at diagrams that show the entire chromosomal layout of a cell, we call that a karyotype. So what you are looking here is a karyotype. Yeah. Now this individual here is a female. You can tell because there are two X chromosomes there and no Y chromosomes. Okay, so it's an actual microscopic image of a karyotype of a female person. Okay. Here's the same thing, but the DNA have been stained, and you can see some of the different properties that distinguish some chromosomes from other chromosomes. So it's really important when we do it to understand so that we can differentiate between chromosomes and their locations, and subsequently understand sort of which gene is found where. Okay, you can see the black sort of shady part in the middle indicates the centromere location. Okay, now you will notice that some of these chromosomes, sometimes the chromosomes appear, let me go back 2 steps, like an X, like that, and then at other times the chromosomes will appear as a straight singular line. Now, when a chromosome sits in this kind of normal stage, it looks like this, and this is a chromatid. Okay, it is only during the DNA replication stage that it forms a kind of an X shape, which is the chromosome, and those two sides of the X are actually the same, and that's during the DNA replication phase. Okay, and so that X shape is what happens when the DNA synthesizes into its replicates, and then during mitosis or meiosis, it will then split up. Okay, so that's the kind of the only distinguish between the two that I forgot to mention in class. Right, this individual is a male with the presence of a Y chromosome and an X right there, and you can see there the stains of the chromosomes show up differently. That's how we can distinguish chromosomes from one another, because the molecules in these chromosomes are all different, because the proteins that sorry, not the proteins, the nucleotides are all different within them. Okay, now humans chromosomes can be distinguished by their relative size. the position of the centromere and the patterns of light and dark bands from the staining. Now, the light and dark bands indicates the differences between the genes within those chromosomes. The different genes have different lengths and therefore come up with different light and dark banding when you stain them. Okay. Human chromosomes are made up of two particular categories. You have the autosomes and the sex chromosomes. Okay. Autosomes are like your normal chromosomes. They are the first 22 matched pairs of chromosomes that are present in both males and females. Everybody has the autosomes, and they can, contain the majority of the information. However, sex chromosomes are the ones that determine the sex of the individual cell. Right? The X chromosome or two X chromosomes characterize a female, and an X and a Y will characterize a male. Okay, the presence of the Y chromosome is what determines the sex of that particular cell. Right? If it's present, it is a male. If it's absent, it is a female. You can see there, the chromosomes from 1 to 23 plus the X and Y are ordered according to their size, and you can see even though some of them are very similar in size, for example 9 and 10, position of the centromere and the banding are slightly different when they are being stained, and therefore we can distinguish chromosome 9 apart from chromosome number 10. Okay, it's ordered according to decreasing size. So chromosome 1 will always be the largest one, 2, 3, 4, and so forth and so forth. Basically, the larger the chromosome, generally, the more DNA it has and the greater number of genes it will then contain. Chromosomes come in matching pairs and are said to be homologous. The position of the centromere is in the middle, and it contains about 1 million base pairs of repeated sequences. Okay, now surrounding this is a protein structure called the kinetochore, which also acts as an attachment site for the spindle fibers during the process of mitosis and meiosis. So that little protein segment there is what allows the spindle fibers to connect, and when they connect, they bond to the kinetochore, and it allows the protein to then split up the chromosome centromere right around the middle, that little pinching part. Okay, so that's just a bit of an interesting thing. There's the microtubules from the spindle fibers, which forms the backbone of your cell, or the structure of your cell, right? When you look at a chromosome, it might look like a neat little X, but in reality, it's kind of a bit like spaghetti, coiled everywhere and dangling pieces all over the place, right? Now, really briefly, looking at sex determination. So, in humans, presence of Y chromosome of the sperm will determine the sex of the individuals. Therefore, males are an XX, and males are an XY, and females are an XX. Humans, most other mammals, some insects and plants will use this particular system, but this is not the case for all animals. Okay, so birds and fish and crustaceans, for example, are reversed. Yeah, it's called a ZW sex system, but it is the presence of a W chromosome which determines the sex of an individual. So females have a heterozygous match. Okay, so they are a ZW, whereas males are a ZZ. Right, so you can see there are slight differences there. Some organisms have an XO sex determination system, where O represents a missing chromosome. So basically, one sex in the population will have one extra chromosome than the other one. So in this case here, females will have one extra X chromosomes, whereas males lack it, and that's found in some insects such as grasshoppers. Okay, the environment plays an important role. Some animals, the environment will determine the sex, and that's called environmental sex determination. The temperature, most commonly happening in reptiles. For example, in turtles, when the temperature rises above 31, you actually get more males, whereas when the temperature drops below 27, you get females. And so, turtles have to do this very interesting thing where they need to get the right height of the actual the nest where they lay their eggs in the sand, so that you get a balance of both. Crocodiles will do the same thing as well, and it's a very complicated process, and which shows kind of the genius genetics behind turtles being able to do that so intuitively, find the right spot, dig to the right depth, and then sort of have it work out in that kind of sense. All right. Now I want to look at chromosomal abnormalities. Now there's a whole branch relating to this called cytogenetics, which is the study of inheritance relating to the structure of chromosomes. Yeah, we can identify karyotypes using a microscope imaging, and you create 15 cells. You can create a karyotype out of the 15 cells, and by doing this, you can actually identify what are some of the abnormalities that occur and how they are linked to congenital disorders. So basically, disorders that are present at birth. Now, these congenital disorders, looking at how they're linked to chromosomes as a whole, is slightly different to looking at how particular genes affect an organism. Yeah? And so what scientists look for in cytogenetics is looking at how the changes in total number of chromosomes, changes in parts of the chromosomes, or changes in the arrangement of chromosomes will affect how an individual is, right? And so that kind of looks at a whole chromosomal level and how that affects it. So we're going to look at a few abnormalities that occur as a result of modifications to chromosomes, not just genes, okay? There are basically 5 things that can go wrong, and you can sort of pick out sort of where along the process it can sometimes go wrong, right? The first one is when you delete a segment of the chromosome, yeah? So if you imagine chromosome 4 is meant to be around about that length, and then all of a sudden, during the replication process, a big segment, a big chunk of it gets cut off and then matched in, all of the genes in there go missing. And when all the genes in there go missing, it creates complications, right? You can also have a chromosome abnormalities around duplication, whereby the same segment is duplicated twice, and so the cell will have issues trying to read which one's the correct sequence according to this particular arrangement. You think it might be a harmless thing, but it's actually not. Sometimes you get inversion where you flip the sides upside down, where if this was at the top close to the centromere, it now gets flipped and it ends up ending at the bottom. And because it's genetic code, it's still read from that particular order, from top to bottom, but then now it's read in reverse, and so it creates a whole new issue because that means it results in different proteins being produced, different regulatory functions of those genes. Sometimes you can get a substitution between 2 different chromosomes, so one segment is removed from one chromosome and attached onto another chromosome, resulting in a change in this chromosome and a change in this chromosome, which can be quite a bad thing sometimes because now you're affecting both chromosomes, not just one. And the last one is called a translocation, where they basically swap segments, once again affecting both chromosomes. Yeah, this segment breaks off, attaches to that one, this segment breaks off, attaches to that one. And this is what happens sometimes when you have that recombination process in meiosis, right? During that crossover recombination, and it does it wrong or it does it incorrectly with the wrong chromosome, this can end up being the result, okay? Depending on the nature of the change in chromosome, you get a different syndrome or a different abnormality, okay? So the most common one we know of is Down syndrome, where there is an extra number 21 chromosome, so it is a duplication of an entire chromosome, not just part of a chromosome, but an entire chromosome. Chromosome 21 happens in 1 every 700 live births, which is fairly frequent considering the population of Australia, all right? There's other ones such as Edwards syndrome, Patel syndrome, Klinefelter. We're only going to look at a few of these, but it is worth sort of taking note as to how these are systematically categorized, okay? Extra 21, in brackets, you can actually identify how that is labeled, yeah? 47 indicates the number of chromosomes this individual will have. So in this case here, because it has an extra 21, it now has 47 chromosomes instead of 46. And the comma separates the individual change from the overall change, yeah? So plus 21 tells you there is an extra 21 chromosome, okay? Extra 13, so 47 again, extra 13, extra sex chromosome, 47XXY or XYY. If you are talking about deletion of a whole chromosome, then you could say it's 46XO, right? Or part of a chromosome, you indicate which part and which arm it's missing from, right? So 46, 4, chromosome 4, P minus is missing from the P arm, 46, 5, P minus, right, missing from the short arm, which is the P arm over there, resulting in Chat and the other ones as well, okay? We're going to look at a few of these. So the ones we're actually going to look at here is Down syndrome, Edward and Klinefelter syndrome, where you have extra chromosome 821, extra 18, and an extra sex chromosome deletions, looking at Turner syndrome and Chat syndrome, okay, when you have missing parts or whole chromosomes deleted. All right, let's take a look. So Down syndrome is probably the most common one that we know of, also known as trisomy 21, tri representing 3, zome for chromosome, trisomy 21 chromosomes, okay? Characterized by mental impairment, individuals with Down syndrome have slight to major mental impairment, and this varies between individuals as well. You can have high-functioning individuals, and you can have then low-functioning individuals as well, okay? So Physical growth delays, so what that means is they don't grow as fast and they grow later, right? Low muscle tone, so they lack muscular features in which you would expect a normal individual to have throughout their lifetimes, and they also have an increased risk of health problems such as heart defects, leukemia, and thyroid disease. Now, the key thing to note here is that whole extra 21 gets read differently, right? And so the cell now has this kind of predicament. You would think that by having just one extra chromosome, the cell can sort of recognize and identify which one to sort of look at. But if you think about the genes that are being expressed in there, it doesn't know which one to interpret, and it doesn't know how to interpret them in a way that is beneficial to the organism, right? And so it creates this kind of big dilemma or problem and confuses the way that the genes are being transcribed and translated, right? And so that is what sort of results in this. Now, when you have a duplication of a whole chromosome, there are significant complexities that are being involved, right? Isolating and identifying how the genes interact with one another for things such as heart defects, cancer, and thyroid disease and things like that become a big problem. And so individuals who have Down syndrome, there's no cure for it, right? There's no easy way to sort of just treat the problem, but really we can only identify the symptoms, manage and reduce those kind of the impairment that it creates to lifestyle, and then just purely from understanding that, we can just do our best to sort of help individuals be able to obtain a quality of life, right? You can't cure Down syndrome because it occurs in every cell in the body. In every cell, there is one extra 21 chromosomes. And so it's not something that you can just cure right away. And so, I think that's, sort of worth mentioning there, particularly if you have, family members that you know of who have Down syndrome as well, and understanding some of these things help us to manage those conditions, right? Klinefelter syndrome, so 47XXY, a duplicated X chromosome, yeah, a whole X chromosome added. Individuals with Klinefelter syndrome are still male because of the presence of a Y chromosome, but they are slightly different, right? Because they have an extra X chromosome, a few different things sort of occur. Generally, males are sterile, they have a weaker muscle tone, have a greater height and a poor coordination, smaller genitals, breast growth, and reduced sexual desires. And so you can see there, X there, and a Y there, right? And so some of the genetics present in the X chromosome gets expressed or overly expressed within the individual, and so there are female traits such as breast growth showing up, a wide hips, long legs, etc. Yeah, all appearing within the individual. Okay, Cri-du-Chat syndrome. So when you have a deletion of a segment of the short arm of the 5th chromosome, 46-5p minus or 5p monosomy, characterized by a low birth weight and poor growth, cognitive, speech, and motor delays. Once again, Cri-du-Chat syndrome can also vary in terms of its cognitive abilities as well. Generally hyperactivity and aggression, and they also have difficulty swallowing or sucking because they are lacking a particular muscle behind the throat that allows the swallowing action to feel normal, right? And that is because the proteins that are responsible for forming that muscle on the 5th chromosome is missing, right? On the P arm in particular. And so that 5th chromosome is lacking a segment of its P arm, and so it's shorter than it's supposed to be, right? Now, sex chromosome abnormalities. So errors can also be made in the sex chromosome, and this usually occurs during gamete formation in meiosis, right? And so what happens is you can get complications occurring where when the cell divides into two, you would hope that you'd be able to, have half of the genes in one and half of the genes in another egg. But when it doesn't, we call this a non-disjunction, yeah? Non-disjunction for when it doesn't split properly. So in this case here, you've got meiosis here, round 1 develops 2, and then round 2 develops 4 individual gametes. In this particular case here, you have a non-disjunction in the second half, right? In which case, rather than splitting all in half, you have 2 chromosomes fitting into one particular cell and lacking in the other. Now, this is just for one set of chromosomes, not for all of them, right? And so what you've got here is a slight disjunction of this one extra chromosome being added into the karyotype of the individual, right? And depending on which one is being added, results in a different syndrome, right? And so if you imagine, XX results in a normal female, XY results in a normal male. If you have double X, you have a triple X female, double XY, Klinefelter syndrome, XXY, but now the X and the Y is here. the non-disjunction occurring in the sperm, then you have also Klinefelter, but of a slightly different kind. If you have nothing, so that other non-disjunction cell coming in from the female parent and the X from the male, then you have an XO resulting in Turner syndrome, the vice versa. If you have an OY, it is a non-viable egg. It doesn't develop, right? That kind of shows you how important that particular X chromosome is, right? And if you have an XYY, the non-disjunction occurring in the sperm here, then you have a double Y male, right? We're only going to look really at Turner syndrome here, where there is a deleted X chromosome, 45 for the total number, XO, right? XO being female, right? Characteristics are generally sterile, webbed neck, low sets of ears, and a low hairline. Because they are sterile, or more today, this is what causes sterility, is that they have no menstrual periods, there's no breast development, and there's a high risk of heart defects, diabetes, and thyroid disease, right? And so that's it there, right? Missing that X chromosome, right? Now, a lot of these things we're just sort of highlighting, getting you guys to understand how the chromosome disorder affects the individual, right? And these are real, real conditions that affect real lives in people. And the more we know, the more we can help, and the more we can help manage these things and work through them. And so it's really important to understand sort of that's the role of genetics, and understanding how genetics can alter lifestyle. We're not just here to do research and just to understand these conditions, but we're here to then, by developing our understanding, we can then change the outcome of the futures of these people, all right? Cool. So that's it. Hope you guys enjoyed that, and I'll catch you guys next time. See ya.